It's time now to see how neurons can be electrically excitable. To say that neurons are electrically excitable suggests that they can generate electrical currents. So we're going to start there. What is an electrical current? An electrical current is the movement of some kind of charged particle. In a wire, it would be electrons. But for living cells, neurons, the charged particles are ions of different kinds. There are sodium ions, potassium ions, chloride ions, calcium ions. But they're all charged particles, and the nature of the movement of these charged particles is into or out of the cell across the membrane of the cell. Now, ions typically can't cross the membrane itself. They need a channel. And throughout the membrane of the neuron are proteins, and some of these proteins are ion channels. So if these channels are open, then that will allow a given ion to move across the membrane. And the channels might facilitate the movement of an ion into the cell or out of the cell. But regardless, a moving charged particle is an electrical current. So the electrical nature of neurons has to do with ions moving across membranes. Okay, so to understand how a neuron can generate an electrical signal called the action potential, we're going to take this cartoon neuron here. Here we have the cell body and then the axon coming off here to the right here. Now in the membrane of the axon are these colored objects, and they are proteins. And there are two different kinds of proteins. The red ones are sodium channels and the blue ones are potassium channels. Now the way they are shown as X's here, they are shown to be closed. So these channels are closed, they're not open, which means that the ions can't cross the membrane. Now we need to know the initial concentrations of these ions as well. As it turns out, the fluid outside of the cell has a lot of sodium, whereas there's not much sodium inside the cell. On the other hand, there's a lot of potassium inside the cell, but not much potassium outside the cell. So there's a concentration gradient for both ions. And if these channels were to open, then these ions would move across the membrane. But if the channels are closed, the ions are maintained at these concentration differences, but it makes the neuron capable of generating an electrical signal. So the action potential, the electrical signal, has its origins right here where the axon comes out of the cell body. And what we'll see is the key step is that these first sodium channels right here are going to be opened up, and that's going to start a chain-like reaction that will move all the way down the axon. So let's see what that looks like. Okay, so now what we see in this picture here is that these first sodium channels have opened up. Now because there's more sodium outside than inside, the sodium moves into the cell. All right. Now as sodium moves into the cell, some of the sodium diffuses a little bit down the axon, and what that's going to do is trigger the potassium channels to open up. Well, remember, there's a lot of potassium inside, not so much outside, so potassium moves out of the cell. So both ions are moving now because they have concentration gradients. They weren't moving before because the channels were closed, but when the sodium channels open up, sodium comes rushing in. That triggers the opening of the potassium channels. Potassium rushes out. Now this sequential opening of channels then continues along the length of the axon. So in the next scene, we see the next set of sodium channels has opened up, and then the next set of potassium channels has opened up. Again, sodium's coming in, potassium's going out. Now notice the original set of channels, they've closed back up again. So this is a one-way domino-like sequential event of ion channels opening up along the length of the axon. And this electrical disturbance will move all the way down to the terminal. Now in this slide we're going to introduce a way of visually illustrating this electrical disturbance. Here we have a section of axon, and we see some of these channels are closed. It looks like the electrical disturbance is at this location. We have the sodium channels are open and the potassium open here, and that disturbance hasn't yet reached this point further down on the axon here. But these green things here are going to be recording electrodes. We're going to insert a little microelectrode to record the charge difference between the inside and the outside of the cell. So this is the interior of the axon, and then this is the fluid surrounding the neuron. Now down at this location, remember the electrical disturbance hasn't reached this location yet, but if we were to just stick an electrode in there, we would measure a charge here of about minus 70 millivolts. Now it's negative, right? So the inside of the cell is more negative relative to the outside for reasons we're not going to get into. But this is called the resting sort of voltage, you might say. And when the cell is at rest, the inside of the cell is simply just more negative than the outside, and the magnitude of that negativity is about minus 70 millivolts. So that's a measure of the charge difference between the inside and the outside of the cell. Now what we want to do is use this kind of graphical representation to capture the electrical disturbance that is the action potential. So now we're going to take a look at what this graph would look like. So this time on the x-axis, and here's the charge difference on the y. What is that graph going to look like over here if we insert our electrode right near the sodium channels that have just opened up here? So I'm going to move this away here. And what we would see then is on an oscilloscope, we would see a positive deflection of the signal, right? So notice this is minus 70 here. That was the resting voltage. And now that sodium ions have come rushing in, then that's going to make the interior of the cell more and more positive. It'll get up to 0, even go past 0. It'll become more positive. So this is part of what we're going to call the action potential. It is the first half, you might say, of the electrical disturbance. See, because now as sodium comes in, remember the potassium channels are going to open up, and potassium is going to leave the cell. So if we take a look at this electrode here, and we'll see what it records, notice here what we're capturing is the second half of the action potential. As potassium leaves, the cell's going to get more negative again, right? So a positive charge leaving the cell is going to leave the interior of the cell relatively more negative. So together, these two halves of the curve are the action potential, and they are capturing this electrical disturbance. The opening of sodium channels causes sodium to rush in, and that makes the inside of the cell more positive, but then the subsequent opening of the potassium channels and potassium leaving makes the inside of the cell more negative again. And we can imagine then that this electrical disturbance, as it moves down the axon, would look something like this. Here we have our axon. Here's our the two parts put together. Here's our action potential signal as measured, say, on an oscilloscope hooked up to our electrodes. That disturbance would move all the way down the axon as these ion channels open up in a sequential fashion. So it's kind of like dominoes. So if these first channels open up, then we're going to get this all or none signal called the action potential that will move down the axon towards the terminal. Now we should say one more thing about this action potential. It has the same amplitude all the way down, right? So it is an all or none signal, and it does not diminish in its amplitude as it goes down the axon. It just looks like this signal, except it's now just going to be measured at subsequent points along the axon. So then the question, of course, is, well, what triggers the opening of these first sodium channels? Because once they open, it's the domino event, and it won't stop. But what is it that triggers the opening of these first sodium channels? And the answer to that has to do with activity of axon terminals that are making synapses on the cell body and the dendrites out here in the receiving zone of the neuron. So you'll recall that neurons don't make physical contact here, but there is a space here, and this junction is called the synapse here. Here we have our axon, and this would be where the electrical signal is racing down here, sequential opening of ion channels and ions in and out. And it gets down to the terminal, and then what we'll see here is there will be some chemicals released here, and these chemicals then can electrically excite the target cell. These chemicals are called neurotransmitters. So let's take a look at what they do. In this slide, you see here's our input cell. The action potential will race down the axon to a synapse, and now we have a magnification of the synapse. So here's our terminal, axon terminal. Here's the synaptic space, and here's the dendrite, or the cell body. Now, notice on the dendrite, or the cell body, are more protein channels, and we'll see how they play a role in a moment. Let's go back to the terminal. So when the action potential comes down to the terminal, that electrical disturbance causes a very important thing to happen. These chambers here, these vesicles, they are membrane-bound chambers, and they contain special chemicals called neurotransmitters. Well, when the action potential reaches the terminal, these vesicles begin to move towards the terminal membrane. And then the membrane of the vesicle can fuse with the membrane of the terminal. And that opens up the chambers so that the chemicals inside, the neurotransmitters inside, can be released into the synaptic space. It's a very, very small gap here, so they simply diffuse across this space in a very short time. And these neurotransmitters are like keys that fit a lock. The receptor proteins here are like a lock, and the transmitters are like a key. And when the transmitter binds the protein, the receptor here, it can open a channel. Again, another idea, the same kind of idea of a channel opening up. Now, when these channels open up, ions that are in the fluid out here can enter the target cell. For example, sodium ions. It's important to note the transmitter itself, the neurotransmitter, does not enter the target cell. It's just the key that opens the channel. It's ions again that come in. And remember, the movement of charged particles is an electrical current. So the effect of these transmitters is to generate an electrical signal in the target cell at that location, at that synaptic site. Here we see another version of this. The action potential coming down here. Notice this is our little icon. This will be our little sort of oscilloscope icon for an action potential. It's one action potential. So again, just to recap, that's a sequential opening of ion channels, and ions, sodium in, potassium out, and it comes all the way down to the terminal. There's a bunch of chemistry that goes on here we're not going to get into, but the effect of the arrival of this signal is that these vesicles containing neurotransmitters are going to fuse with the terminal membrane. The transmitters get dumped out into the synaptic space here. They diffuse across this space, and they randomly bump into things, but if they find their target receptor here, they can open it up. Notice this this receptor is closed. It doesn't have a neurotransmitter binding here. But here one has opened up the channel, and now what comes into the target cell are ions. Now, if there's sodium ions that come into the target cell, that tends to excite the target cell. If this transmitter opens up a different kind of receptor and allows negatively charged ions into the cell, that tends to inhibit the target cell, so make it less likely that it would generate its own electrical signal. So what's happening at synapses here then is the release of chemicals is causing a small little electrical signal at that synaptic site in the target cell. Now we're ready to see how activity at synapses can trigger the action potential as an output signal for a target neuron. So here's our target neuron again, and remember, it's these important sodium channels right there at the beginning of the axon. If they get opened up, then you'll get an action potential. So the question was, when do these things open up? And that has led us to the answer, which is when there is sufficient activity at synapses on the target cell itself, or out on the dendrites. So these channels are going to open up when a threshold amount of sodium enters at synapse sites. So here we have a synapse. Here's our terminal, and the action potential has come down here from the input neuron. Neurotransmitters are released, and here the channels are opening up, and sodium comes rushing in. Now, this neurotransmitter might open up this one. More sodium will come in here, right? So the more transmitters released here, the more channels will be opened up, the more sodium will come in. If a threshold amount of sodium enters the cell, that is sufficient then to trigger the opening of these first channels, and then you'll get the domino event going all the way down the axon. So that then connects up this idea of the output signal. How is the output signal initially generated? Well, it's the result of activity in the receiving zone of the neuron, either on the cell body or the dendrites. It's the result of activity at synapse sites. So neurotransmitters can cause electrical signals at those local regions, but they can build up to reach a threshold, and so then you get the triggering of those first sodium channels. Now let's take a look at what that looks like in our little graph. Remember, minus 70 is the kind of the voltage when the cell is just resting. It's more negative when it's just hanging out. And the idea is this threshold may be maybe 5 or 10 millivolts, right? So if enough sodium enters at synapse sites all over the neuron, if enough sodium enters so that the cell becomes just 5 or 10 millivolts a little more positive, then that triggers these first sodium channels, and that will then send the action potential on its way. Remember, the action potential has the sodium coming in, that's the first half, and then potassium leaving. And this whole signal is going to move down the axon. Once it gets going, it's the same amplitude, and it just moves all the way down. It's an all or none phenomenon. What triggers the action potential? There must be sufficient activity at synapses to reach a threshold amount of positive charge inside the cell.